Okay. Hi, everyone. I hope you can see and hear us well. And let's begin this session of the Bioinformatics Club seminars. Well, I will start with a brief introduction into the idea behind these meetings and the topics of this particular series. Then we'll jump right into the talks and the discussion for today. So yeah, this is the third session of the Bioinformatics Club seminars. And the main idea of these seminars is to have an open discussion of the trends and bioinformatics and computational biology, of best practices, of do's and don'ts. And we have actually already had a couple of sessions previously, one dedicated to the reproducible research methods and tools, and another about the best practices for genetic variation analysis. And we're actually going to continue along these lines, discussing various specific areas of computational biology, of bioinformatics. But then we have all seen the rise of artificial intelligence, and that actually prompted us to postpone the discussions of more specific topics in bioinformatics in general, and actually make the third session fully dedicated to artificial intelligence and its prospects in bioinformatics and challenges associated with its application. And for this session, we have 4 meetings in our schedule right now. And these 4 meetings will take place on Tuesdays at 6 p.m. Central European Time, starting today, and then repeating every 2 weeks. So we have the first meeting today about the genetic variation analysis. Then in 2 weeks, on August 19, we'll have a discussion about gene expression data and transcriptomic studies. Then we'll have a third meeting about proteomics and about protein structures on September 2nd. And then we will conclude this session by a more general discussion about AI methods, about the challenges and perspectives. We'll have, I hope, a good technical talk about various new approaches to developing artificial intelligence methods and to applying these methods in real practice. And before we begin today's talk, I'll spend just a couple more minutes introducing you to the motivation of these meetings. So one of the key questions which I think many people have now is whether or not this rise of AI methods will actually lead to a dramatic change in the way we actually do our analysis and the way the bioinformaticians actually do their routine work. And this is actually one of the central issues that we have. And in the past few years, we have seen actually quite a significant shift from just the first versions of ChatGPT, which were obviously very fascinating in terms of that it was capable of producing very coherent, very nice outputs and actually chatting like a real human in many cases. But still, it actually was limited in a lot of aspects. But in these 3 years since, or 4 years since, I believe everyone has known about ChatGPT, there have been a lot of developments, and in particular in the field of developing so-called artificial intelligence agents. And this is one of the things which actually prompted me personally to organize this series of seminars, because for those of you who are not familiar with the terminology, basically an AI agent is kind of a system that can solve various tasks beyond just providing an answer to a user question or prompt. So typically, this is a sort of a combination between a traditional computer program in which you have execution of certain certain tasks or certain functions in a particular order, but actually a more complicated thing when you use artificial intelligence models, not to give a single response to a single question, but actually to approach some step-by-step analysis. And in the world of programming, there are actually already agents that can handle the development of the computer program in a single run from just the user's description of what the program needs to do. So the idea there is that the AI agent not just creates the code, but actually creates the test data, runs the generated code on these test data, evaluates the results, and does the iterative fixing of issues and repeats the testing until it reaches the final result. Well, essentially just automates the typical software development pipeline that is typical, again, for software developers in software development companies. And if you just Google sort of the AI agent in bioinformatics, you will see a very large number of studies. Just one of these is on the right-hand side, not specifically chosen for some particular reason, but just because it has a catchy name like AI Hope. This is just one of these AI agents which are starting to appear in the field of bioinformatics, which can actually automate the routine work of bioinformaticians when you just do the natural language prompt. And the agent actually does all of these sort of stuff that we bioinformaticians typically do, meaning gathering the data, aggregating the data, performing some of the basic analysis and some of the like routine analysis tasks, running the pipelines, etc. Again, there was a lot of development in this field, and for those who are interested, I think that we'll have a more detailed discussion of agents in the concluding meeting of this session. But I think that the rise of the agents in many people actually generates this condition, which is called in some cases, you may hear this phrase AI anxiety, which is basically this sort of psychological attitude towards AI in which people see that the development of AI models can actually lead to massive displacement of people who are doing the work, in particular in bioinformatics, people who are doing the analysis may actually get replaced by intelligent systems who are doing this analysis. And there is a lot of debate about whether it's true or not, whether these systems will actually do this replacement or not. And this is exactly one of the things which I want us to discuss during these meetings. I want us to have a discussion of how do we apply the artificial intelligence methods now, what is their scope, which tasks they're good at, which tasks they're not so good at. When I say artificial intelligence, I do not only mean large language models or LLMs, I also mean some of the state-of-the-art neural network architectures, which are not, strictly speaking, large language models, but can still be called AI, generative AI in particular. And one thing which we'll try to one question that we'll try to answer is, what is the proper role of the current methods in the analysis pipeline? And the second question, which I would really like every speaker to address during this session, is whether or not we can actually expect these agent systems and the progressing development of AI actually lead to the massive displacement of bioinformaticians, or how would they transform the routine work that each and every one of us is doing in their particular field of research? And that's actually the second question, which I believe is very important and which we will be trying to address during these meetings. And without further ado, let me introduce the first speaker for this session. Many of you, if you have already attended our previous meetings, have already seen him. It's German Demidov from the University of Tubingen. Hi, German.
Hello, everyone.
Yeah, I think I can just stop sharing my screen and pass the microphone to you for your talk. Now we'll have a talk from German. After that, we'll have some discussion of some specific questions related to the talk. After that talk, we'll actually have another sort of brief talk from my side, some of the interesting aspects which I would like to add to the discussion, and then we'll have a more sort of generic discussion of what is going on. So yeah, German, please, the floor is yours.
Thank you for the nice introduction, Yura. You can see my slides, I guess. If don't, please let me know.
We can.
Perfect. Yeah, so yeah, I will be talking about the AI analysis of genetic variation in terms of clinic, in terms of genomic medicine. I work in the University Clinic Tubingen, and for most of people, Tubingen doesn't mean anything. Just a small German town, 90,000 people. But for some reasons, it's first of all, it's a university city, and for some reasons, there are two really strong sites in this university city. I mean, scientific sites is machine learning and medical genetics. So everyone is really surprised that medical genetics is such a big topic here. So I will be discussing this from the perspective of like a really large center which performs a lot of analysis, mainly for rare disease patients and mainly for cancer patients. Yep, I managed to switch the slide. Perfect. I'm still struggling with PowerPoint and using ChatGPT or something. Yeah, it it makes me scared. This whole new technology. I'm joking. I'm using ChatGPT, as probably all of you also. The disclaimer. So we have the two questions to answer. Can we trust large language models, and would there be a need of mathematician to perform routine genetic analysis? I will be trying to answer in 20 slides approximately, so it will be a short presentation, hopefully. The talk is addressed to general audience. I will not discuss architectures of neural networks. I couldn't discuss even if I wanted, because it's not my primary topic of research. I'm using the tools, but even in our lab, there are people who are doing it at the higher level. I'm just doing the general overview of people do here in our institute. I will address the question 2 immediately. Would there be a need for mathematician to perform routine genetic analysis? We developed a mathematic pipeline for performing routine clinical analysis, and there is a button, and the button is like analyze everything. So there is this thing, and every medical doctor, every person without any education in bioinformatics can press this button and run the analysis. But yet there are only in our group, there are several groups, there are around 20 bioinformaticians. So yeah, there is still a need for bioinformatician. And yeah, I strongly believe the ChatGPT or any other GPT agent cannot really replace us, but maybe it's a religious belief. It's up to you. In the end of my presentation, I hope everyone will decide. First of all, one more disclaimer. I'm making many disclaimers. I was always thinking that AI is something like really high level, like something which is. I was reading about this strong theory of artificial intelligence, and for me it was always something very advanced. But I was at the talk, and the person was doing something very simple, like a linear regression analysis, and he was calling it AI. And I kind of silently complained to my colleague next to me, my senior colleague, and they told me that it's actually AI. So if we, this is definition from Coursera, one of the, I would say, it's kind of, it has the authority, this resource, and there are many other resources which make this diagram that deep learning is kind of encompassed in machine learning, and it all contains in artificial intelligence. So technically, if you take estimate something as the mean, it's like linear regression, performs simple linear regression, you still do AI. We will not be talking about AI in this sense. We'll be talking about deep learning in this talk. At first, several papers which kind of turned our field, this medical genetics, which changed our field in the recent several days. This is not a journal club, so I will be focusing on the experience of our institute, but I must give some brief introduction in what changed our view on the medical bioinformatics. First of all, it was AlphaMissense. I will just briefly describe it. It's basically takes the first same steps, a bit improved from AlphaFold, which got a Nobel Prize, I think, in the last year. and perform very similar analysis, and then it assigns scores to the variants, to all the missense variants. Is important because there are variants which are not only missense, to the missense variants, if they're pathogenic or not. And immediately when the paper was published, we made the emergency journal club session about this paper. We invited people from diagnostics, which is super rare. Normally they are busy people, and we don't invite them for our bioinformatics journal club. But in 1 week after the paper was published, we had a journal club, and we realized the huge potential behind this. Normally, the medical doctors don't really trust our computational predictions. These CAT scores, PhilOP scores, some other scores, they know that they are not really good. And in previous times, like when I just joined our institute in 2018, it was like they never really paid attention to them. And with AlphaMissense, I would say it changed. So with AlphaMissense now, they really they can really insist on the analysis, on the continuation of the analysis of some missense variant, which is just a high computational score, and without really strong other indications. So this tool changed a lot. This is the one picture from there. It's based on multiple sequence alignment. We mask some positions, then we run the evoformer. It's the key element of AlphaFold. And instead of trying to predict structures, now we predict variant pathogenicities here. And it was tested on the known databases of pathogenic and benign variants, which is ClinVar, and also so-called protein gym. This is a technology for mass testing of variants, of effects of variants. It's very noisy technique, but still you can see correlations there. Yeah, nice paper, and it was immediately introduced in our pipeline. We immediately annotated in 2 weeks after the paper was released. We immediately annotated our new results with AlphaMissense, and we felt that it led to a change in the how medical doctors, how genetic analysts, how they treat these computational scores. Second paper that really changed a lot for me in particular, I was doing a lot of so-called burden tests. It's the tests when you compare cases and controls, when you merge rare variants. It's for common variants, you can use GWAS, but this is kind of something for very rare variants. Instead of doing GWAS, you collapse variants by genes. So you take, for example, all the loss of function variants in cases and loss of function variants in the controls, and then you compare. And if it's significantly bigger in cases, then they may say that this gene is maybe playing some important role. All good, but there are many variants in the genes, and to collapse them in a smart way, typical strategy was just to choose the qualifying variants and record them 0 or 1. 1 is if the patient has the qualifying variant, 0 if the patient doesn't have qualifying variant. What if the patient has several of these variants? What if the patient has variants with high CAT score or high AlphaMissense score? Doesn't matter. The typical methods for burden analysis, they allow usage of 1, 2 covariates, not more. And this particular deep set network analysis, the work published in last year, it was also quite changing. So they basically took all the phenotypes from UK Biobank, if I'm not mistaken. They annotated them with all different types of annotations, as you can imagine, like even these CAT scores. I think AlphaMissense score was also there. And then they ran of course, they did the embeddings of the variants, and then they ran some sort of this deep set architecture network, and they got the gene impairment score in the end. And yeah, I think first, it allows, it has much higher power in detection of genes responsible for phenotypes, and also it can be incorporated in the polygenic risk scores. You can incorporate rare variants into the framework, which is typically used only with common variants. So very cool paper. I recommend to use it. Not to use it, but to check it out. And the third paper is the last paper I will be talking about. It's again, I mainly work with rare disease patients. We also work with cancer in the institute, but my main specialization is rare disease. And this is one of the really cool papers. They use few-shot learning or even zero-shot learning. What does it mean? You have very tiny amount of the examples there, and they are of course unlabeled. And the goal is to diagnose these patients with super specific phenotypes, which are seen maybe once on Earth or once in the described universe of rare disease patients. It can be a patient with like 2 genes affected or 3 genes, so it's like a mixture of different symptoms. And this particular tool is supposed to solve this problem just by looking at similar genes, similar samples. So this is how it looks like. I suggest to look as normally, like first panel A. It's first you need to create the knowledge graph. So this is prior knowledge. You connect genes, you connect phenotypes from literature. You mine literature for this, and then you remove the edges in the graph which are not connected, and it simplifies the whole thing. Then you let's look at panel B. You input candidate patient phenotypes, genes phenotypes, like all the phenotypes which are described in HPO terms. I think I don't have time to really describe what HPO terms is, but like, let's imagine the whole human phenotypes can be described in the tree-like structure. So the most common is on the top, and most specific phenotypes, I don't know, like 6-finger polydactyly, it can be on the very bottom of this tree. And this tree is allowing description of any complex human phenotype. I generalize, of course. There are problems with these HPO terms, but this is widely used format now. I would say like almost all the genetic medical genetic facilities which work with rare disease patients, they use HPO terms now. And then they train the tool. They simulated the rare disease patients, because rare disease patients are actually rare, and it's they had to test. You can see the test sample that they use. It's a panel D. There are like several thousands of them. So it's not a lot. So they had to simulate the data. They did it in a smart way. Nice tool. Nice approach. Zero-shot learning. This few-shot learning. I think it can be used in our field. That's it. Now we're switching to the large language models and how we are using it in our institute. First of all, this is just the example how I first faced usage of large language models in my practice. I used it as bioinformatician. I was asking it how to code, like how to like typical automatization of my typical bioinformatic things, like how to, I don't know, use find command in this directory and select files with this regexp. But I was giving a workshop in München. It was a workshop for the early career rare disease researchers, and I was showing them how the medical diagnostic of rare disease patients works. And it was the third case that I was presenting. I was going from the more simpler cases to the more complicated, explaining in practice how it works. So I was giving them genomic sequencing of a patient, and then I described the phenotype to them. On the right, you can see the picture. You can see the very broad distance between the eyes. You can see that there are many features. The nose is like depressed nasal bridge. And one of the key things for the medical genetic analyst is to be able to work with these phenotypes. Because you need to match the candidate gene with the patient phenotype, if it's a good explanation or not. And I was asking people, what are you going to do? Like with this description, where are you going to look for the causal variant for this particular disease? I simulated the variants, of course. One may ask, like, why did we use this picture? The usage of patient picture is very sensitive topic, and they usually give permissions to publish their pictures if it benefits to other families. But like using them in the study material, it may be a questionable topic. So it's not a real patient. It was also generated by the neural network. There is a tool called Gestalt GAN, which helps to obtain pictures, portraits of rare disease patients with special features there. So we can use it, for example, for the seminars. This particular disease was not really well represented in this neural network. You need quite a lot of examples, and this is very rare disease. So I had to select out of 20 pictures one which looks like a real patient there. Others were kind of, so this is another application of neural networks in our practice. I was explaining to the people, this workshop how to do the analysis in the traditional way. So there are ways to match between the gene phenotype and the patient phenotype in terms of HPO terms. So you compare two sets on the trees and you measure some distance. I was explaining them what is Resnick distance, semantic distances, and I showed them the tool, so you can put the HPO terms in the tool, and it gives you top 10 candidate genes. And the like the hint that I gave to them that the gene we are looking for, it's in top 10. And then one of the students said, like, I know the answer. I know what's the variant was causal for this patient. And I like, okay, someone did it really fast. And I asked the type of variant because I simulated the variants and I know it's for a specific type of variant. And the patient says, like, not the patient, the person there says, like, it's inversion. It's inversion affecting some gene. And I immediately understood that the person guessed it right. And of course, I asked, like, immediately, you need to check 10 different genes in this case. And it was one of the last genes. So it was just impossible for someone to go through these genes so fast. And they told me that they just simply put these phenotypes in ChatGPT, the whole patient story, and they gave them the really most expected candidate gene, and they didn't listen to my explanation about the semantic distances at all. They just, and of course, I asked the person if this person is professional genetic analyst. No, it was just a person who was biologist and was doing it first time in their life with just ChatGPT. Allowed them this power to do diagnostics out of the box. I would say I was really shocked by the whole thing. I still finished the seminar. I didn't cry. I didn't fall into depression. So it was just the first example, very striking. These are two people who are doing research with these large language models in our institute. So I took the slides from them mostly. I did a bit of my things, but the research is them. We are doing maybe applications of this, but research. So first of all, this is how the diagnostics of genetic diagnostics works. We can start from the left bottom corner. We take there's a patient with suspected genetic disease. Then they take blood or saliva, doesn't matter. Make sample preparation, run sequencing on one of the platforms. Short read, long read, doesn't matter. Then it goes to IT infrastructure. It's typically underestimated factor. We're a very large center. So we have really petabytes of storage there, and the IT infrastructure is one of the most costly elements of the whole thing, of the whole diagnostic facility. So sequencers are expensive, but our bioinformatics is also quite expensive here. And where AI can help us? AI can help us exactly here. After we do bioinformatic analysis, we show this through some decision support systems in some genome analysis software. And genetic analysts are working with this analysis software, and there the AI can help them. They can reduce the time they need to find the causal variant or to find no causal variants. So we should understand that we are here. We are helping to reduce the time. That's how the LLMs are, in theory, should be applied for rare disease diagnostics. There are other applications where AI can be used. In pink, I showed the ones where LLMs can be used. And in just normal black is where deep learning can be used, but not exactly LLMs. For example, pathogenicity classification, it's more complex process than just AlphaMissense. Just pathogenicity score is not telling us whole picture. There are other criteria. I will talk a bit about these other criteria. And LLM can apply these other criteria instead of a person, of course, and of course, save time. Gene phenotype matching, I just described the use case. It was a huge list of different malformations, different issues that the patient has. And then to find the candidate genes is actually costly work. It requires a lot of specialized tool and the brain thinking of a very trained specialist, and LLMs can really save time. Report generation. Report generation is a complex document. We'll talk about this in detail. It's something which is directed to the medical professional, but also to the patient. So it has to be not just containing the true information, but also do it very clearly, and it's very specific. So LLMs can theoretically be trained, and we'll talk about the potential issues in the end of the talk. In the cancer, in the so-called precision medicine, what it is, it's typically when the patients have advanced tumors which don't respond to the first or second line of therapy, they usually undergo the sequencing of tumor normal in our institute, in some places just tumor, and then looking at the variants, looking at the codal variants, they try to find the targeted approach for each tumor here. And of course, we can classify oncogenicity of variants in the tumor to distinguish between passenger mutations and the driver mutations. Then we help select treatment and, of course, generate reports. Prognosis of immunotherapy is typically approach done not with LLMs, but with like more easier techniques, like I don't know, like lasso regression. It can be done. And the targets for mRNA vaccination, I will not be talking about this, but we can also use deep learning for this. This is the schematic description of what kind of data we are working with. We have short nucleotide variants. The short version is S and Vs. We have genes. We see exons here, and these junctions between the exons in the center of the screen are these RNA junctions. That's how the splicing works for this particular isoform. There are indels, which are short insertions and deletions up to 50 base pairs in the body of the gene. And there are structural variants, which are longer than 50 base pairs. There are different data like damage scores, databases with variants which are known to be pathogenic. There are different phenotypes, evolutionary conservation, population allele frequency, everything all together. And of course, it's very complex, and we can use LLM to understand all of this. This is how this is a table person who is doing medical diagnostics should work with. So there are criteria which increase the evidence of pathogenicity of the variant, or which are more like evidence of benignness of the variant. And they have to apply these different criteria in order to cross either pathogenic evidence border or the benign evidence border. And LLM, like trying to apply some traditional deep learning method here, not LLM-like, would be quite a mess, I would say, because the type of data are very different here. And the LLM, in this case, it can work in the sense of understanding different types of data. Understanding, of course, like not literally, it's not understanding anything. LLM is just, it's a stochastic sampling, but still it can be interpreted by a human in a good or bad way. This is GSVAR. This is our decision support system. This is where AI is supposed to be applied. We're working on applying these large language model methods and other like deep learning methods here. What is this? This is one patient, one sample opened in this decision support system. We can see the variants, like Excel-like table. There are different types of annotation in the columns. We can see the chromosome start, end of the variant, reference variant, similar to VCF, but with some additional things. So VCF was not really compatible format to work with. Someone who worked with VCFs understand this, that VCF as a table is not this thing that actually works. And this GSVAR format, it was done to overcome these limitations. And yeah, there are different types of variants, repeat expansions, structural variants, copy number variants, and there are different filter strategies. And previously humans were doing this, and now we are hoping that the LLM will help us to do this. That's how the scheme of variant prioritization using large language models works. It was a tool developed several years ago, and it was not supposed to work with LLMs in the beginning. It was just pathogenicity classifier using random forest. But in the end, we realized, not we, but Dominic, I already showed his picture. He was working into implementing GPT engine in this model. What's happening here? We have variants, we annotate the variants, we take patient information such as phenotype there, and then we use the different models of prioritization. One is the machine learning one. It's a random forest model. quite clear. You take variance from CleanVar and you train a random forest using annotations. Evidence model. What is evidence model? It's a bit more spiritual thing. Evidence model was developed by one of our software engineers, and it actually works really well. So you add 1 point if the variant is located in sensitive domain, for example, and you add 2 points if it's super rare variant. If you add like 5 points if it's de novo variant. And surprisingly, like these numbers, I'm not sure if there is any serious machine learning behind. I would say it's more like intuition. So sometimes humans can surprise us, and sometimes humans can develop really nice, useful models even without the machine learning. I never asked in details how exactly it was developed. I just know that it works. And the random forest model, it's outperforming the human developed model, but it's much less interpretable. It's very difficult to interpret. And the model that was developed by a software engineer, it works really well, and it's explainable. You see why you have these points. And after these prioritizations, you take all this data, really all the data. You don't put VCF on the ChatGPT, of course. You take only candidate variants. If you try to put VCF in ChatGPT, I don't think anything good will work, because the context length, it's still like several kilobases. You cannot put, and the VCF files are, they're huge. So you put everything in ChatGPT 4 in this case, then you use some meta model on top, and then in theory, you should generate patient report here. This is the example how it works. First, you select top 10 genes of both models, top candidate genes for this particular patient. You use random forest model, you use evidence models, then you select variants with some impact there. You, of course, use phenotype, age, diagnosis, many clinical things, and you put everything in ChatGPT. In this case, it was 4.0. Yeah, sorry. It's really terrible how they name their models. I'm still, it's like a mystery. This is the example. A female rare disease patient of age 0, like neonatal, has the following symptoms: facial hemangioma, status epilepticus, causal variant in which of the following candidate genes would best explain these phenotypes. Candidate genes. This is list of candidate genes, and then it's very important to say please, because if we don't say please, the machines will kill us after they capture the world. So always use please. Please provide sources. And there's actually a legend that if you add please, it works better. I really don't know. I think it's maybe not true. I'm asking ChatGPT directly. I'm very like that. But in this case, we use please, and then it actually responded to our request, and probably because we say please. Then it provides the whole explanation why it would be this particular gene would be the most important. And yeah, so it works. It works very well. What's next? It seems obvious, and I think a lot of people are doing the same thing. There is also a lot of people develop custom language models, and we are also doing in kind of I don't know. Is it correctly say in collaboration with NVIDIA, or just with permission from NVIDIA? I don't know. So basically, we use NVIDIA models, large language models, instead of ChatGPT. And what we're trying to do, we're trying to cut the unnecessary information which ChatGPT has, and sometimes it switches from the because of the mechanism how ChatGPT works, which is stochastic sampling. Sometimes it switches from one topic to kind of another topic. It borrows some information from another topic which is not directly related. It's just similar in some context. We think that specialized large language models would outperform the more general large language models. This is our theory, and we are checking it. So these are the models which are available to everyone. And I think you can train it yourself. You can train on top of what is there. And this is the performance so far. This is what's here. On the bottom is rank of causal variant. Closer to 1 is better, which means that the large language models guess the best candidate immediately. If the causal variant is second, that means that large language models first said like about some non-relevant variant, and this in the second attempt it tells you to check the second variant, which is actually causal there, and same so on and so on. Closer to 1 is better, and on the y-axis is covered sample in the process. More is better. So if more samples have causal variant as rank 1, is better. So in this case, we can see that so far it was our Dominic development from 1 month ago, I think. So far, GPT-4o is outperforming our llama-based models, but we are working on it, and we really believe that specialized will be better in our case. This is more or less it about the variants, and a couple of slides about the different tasks. It's not exactly bioinformatics anymore, it's generation of clinical reports. These are documents, and our medical doctors and genetic analysts, they spend a lot of time describing everything in the these reports. Many things are automated here. So many things are already done in kind of semi-automated way. It just takes things from the variants where clinician clicks like put this variant in the report. But many things are still not, and we hope that large language models will help us a lot. It's very, as I said, it's a complex document. It's not a novel. It has to satisfy several important criteria. Not bioinformatics. It's not bioinformatics, it's a medical thing, but it's still very important for us. So first question that we asked in the very beginning, can we trust large language model generated clinical reports? So there are two situations. The one on top is like, you can give this patient report to the patient without reading. It's just you trust it 100%. And the second situation, you have to write everything. It's just a waste of time. You spend time reading the errors that the large language model generated for you. And it would be the easiest for me to say we're somewhere in between. And it's actually true, but the answer is not informative. So yes, we are somewhere in between, but we are in between. I just want to say some words about the practical complexity of this task, because document is complex. First of all, on top you can see happy doctor, because it doesn't have to write patient report. But in reality, it will not be a happy doctor. The doctor will be unhappy anyway, because if they have to write, if the machine will write medical reports for them, why do we need medical doctors in this case? Yes, on the bottom you can see the glitch. I kept it on purpose. I know what I have to do. I asked him like, rewrite this picture, but make both doctors sad. And you can see it. It basically invented the second arm for the doctor. I kept it on purpose. I generated this 1 week ago from the paid ChatGPT. So I used kind of top-notch current large language model, and it still did this. I know what I would have to do. I just asked to regenerate them, and it will just regenerate it with like correct number of arms. But the system needs me here. And I think the same will be, at least the same will be about the report generation. A bit nuanced discussion. This is last slide, I promise. We are almost, I'm almost out of time. So it's the report for the patient should contain not only true information, but also it should be easy to understand, because not only medical doctor reads these reports, also a patient can read the report. And I had experience. I was helping refugees to understand their medical genetic diagnosis. So they were going to a medical doctor and they were receiving some patient report, and the doctor was explaining to them what's happening with them. And then I just said like, I'm a volunteer, you can ask me if you don't understand something. And they were asking me questions, and I realized that they don't understand what the doctors were telling them and what was written in the report. So it's actually a very serious problem that doctors think that they understand something very clearly, but it's actually not. If you use LLM, I think it will be worse. The whole situation will be worse, because LLM it doesn't really have any empathy, any understanding of patient. It can, we all know this ChatGPT empathy with the smileys and something, and we can read that it's very empty behind. I don't think it can help for patients to understand what's happening with them. And even this report, it's not only in personal talk, patient to doctor, it's also in the report. It should be there. It also should contain all the necessary information. So a couple of times I was asking ChatGPT something, and it provides me like, this evaluation thing, evaluate which answer is better, left or right. I was asking complex medical genetic questions, and I like them both because they describe different sides of the same problem. And giving one of these answers would not be enough for the patient. So it should contain everything, but it shouldn't be excessively long. No one is going to read. I made a genetic analysis in commercial company a long time ago, and I got, I don't know, like 50 pages report about all the variants that I had, and I didn't read it. I read only like 1 page out of it. So yes, it's very easy to ask LLM to describe everything about the patient's variant, but it's not useful. This the issue here is to train such a large language model, because someone has to select the better answer and the worse answer, and who can do this? Only very well-trained professionals whose time costs a lot. And if we need a lot of the examples, positive examples, negative examples, we need to this whole project would cost really tremendous amount of money. So I don't really see that large language model would generate the report from beginning to the end. I see that we will be generating maybe some pieces, some parts of it, and it will be assisting the doctor. But at least medical doctors shouldn't be afraid to lose their job. That I cannot say about bioinformaticians the same, but we are here so far. All the automation didn't kill us, so hopefully it will not kill us in the end. And I think it was my last slide, and thank you very much. And I hope you all have a lot of questions, so I'll be very happy to answer them.
Thank you, German, for a very nice presentation. I encourage everyone who has questions to actually drop them in the chat. If you would prefer to unmute and ask the question with your voice, you can do that as well. But I think the chat would be a little bit of a preferred way for us, just to ease the moderation of the discussion. And maybe using my power as the moderator, I will start with the first question. So when you were talking about the power of LLMs to actually do the variance interpretation task, I think that there are actually some other platforms that are already published on, and there is, for example, there is the tool called varchart, which obviously does not do the full interpretation for you, as far as I understand, but it can, I believe, provide quite good responses in terms of the evidence, etc. And there are, I think there was one more tool which I've seen, which I think it was called Maverick. And I'm not sure about the 100% about the name. So now the question is whether you've actually looked into the available tools that already use some of these, like LLM or generative AI transformer-based models for variant interpretation. What do you think about their performance?
Yeah, indeed, I worked. Just short side story. There is such thing as called diagnostic hackathon, and it's very international event where like 100 of experts from different countries come to one place, and there it's not only we're trying to resolve some, I don't know, 10, 20, 30, 40 rare disease patients which didn't find the diagnosis. And it's not only the thing for diagnosis, it's also for the companies who produce bioinformatics software to showcase their recent development. So indeed, I worked with several AI-based decision support system there. And yeah, I like them. Yeah, what can I say? I didn't measure them. I didn't benchmark them. For that, I think the companies, most of this software were proprietary. So most of the software probably will never be published in the open like academic journals, but it's up to the authors to benchmark. I like these systems. It helped with like small team of several people with me participating to solve one patient there. The system correctly predicted the gene which was overlooked in the first analysis there. There are solutions and they work. And if you have a license for this, if you have open source, I think it's very useful to use them.
Thank you. Then I will start reading the questions from the chat. So the first question is whether you actually, when you are describing how you put the information about the patient into the ChatGPT, whether you actually do put it literally into the ChatGPT, and whether it actually is not in violation of any of the European Union restrictions and regulations.
It's a very serious thing about privacy. That's why, of course, the large language models which you use locally is much better. You may provide more data, but no, we use like patient phenotype is not identifying the patient. So if you put particular genetic variants, rare genetic variants in ChatGPT, then it indeed can maybe lead to some clashes. But if you could see in the example that I showed before, we already give them summary of the previous models. We don't give the particular variants to the ChatGPT. We're giving the summaries. So in this case, the patient is not identifiable, and thus it's not violating any privacy rules. But for local large language models, you can afford more freedom here. So yeah, it's a very good question. Yeah, makes sense.
Thank you. Then the next question is, I believe it's a little bit outside probably of what you do typically in your research, but still the question is, how do you evaluate the application of LLMs for gene phenotype mapping in multifactorial disease?
I would say there it's much more complex. Because, for example, cardiovascular disease, there are panels of genes which are known to be connected with cardiovascular disease. And I think there is no specific phenotype that you can ask ChatGPT, can you guess which gene is the causal for this patient? You cannot ask ChatGPT this because the phenotypes are very overlapping and the penetrance of the whole thing is not 100%. So for multifactorial diseases, I would say the applicability in this sense that I described before, matching the phenotype and the gene, finding causal variant, I would say it must be much less in this case. Maybe some different, if you give different input and expect different output, maybe with some different approach, it can be used. But the approach that we use for rare disease is unapplicable for multifactorial.
Okay, thank you. I think there are some more questions in the chat. Please give me a minute. There was actually, I believe, one, not a question, but a remark regarding the benchmark that was made for medical and biological article information. And I think that there are links already in the chat. So for those of you who are interested, you can look into the chat and see actually some of the links from the participants. And then I think that there is actually another question about the rare disease case. So actually, I think the question from there is that epilepsy and SCN1 is very simple, and it will actually be prioritized in the candidate gene by any system. Do you actually have a very rare gene covered only by artificial intelligence tools? So I think the question here is really whether the example that you probably used was I'm not sure whether the epilepsy and SCN1 is the only case that you've shown. So I don't know whether you can comment on that, but
yeah,
that's probably...
Yeah, it was the only case that I showed.
Okay.
I disagree that something is very simple. We are preparing currently a paper about the analysis of epilepsies, and there are many complex things which may happen there. So there are issues, and I work in collaboration with this European reference network, EpiCare, the care for epilepsy patients, and they are high class professionals, and they still struggle with many diagnoses. So many patients remain undiagnosed in this case. So was it very simple? Depends on the variant which was there. And the second, I would say ChatGPT does know anything which humans don't know in this case. So if in this, we are trying to apply ChatGPT to reduce the time for the medical specialist for checking the case, because the time of medical specialist costs a lot. And we have a really backlog of rare disease patients here. So we would really like to work faster, but we just cannot because of all the issues. So LLM can reduce the time. And this like finding some gene which the human didn't find, it may happen, but for that I probably should readdress you to the paper that we published recently. It was analysis of undiagnosed cases, solver-D cases. So there are hundreds of patients which were solved by reanalysis because the variants were overlooked. And I guess it's applicable for this question too. So LLM would find these overlooked variants really fast. And there are examples, yeah. We didn't apply LLM directly in this particular paper, but it would help there. But ChatGPT doesn't know anything outside of human knowledge so far, so it will not find some diagnosis which all the humans in the world would miss. I don't believe in this, no.
Okay. Yeah, I think that there's one more in the chat. Please give me a second. Give me a second. Yeah. So yeah, we have another question related to. Okay. So, okay. So I think that okay, yeah. So I think yeah, there was a lot of wrote a long question, but yeah, I think the question that the question here is what are the main further ideas for improving the llama model? So what aspects is the current research focused on? There are actually several questions here, and okay, are there some surprising results and pivotal points in this work so far? Yeah, I think this rather a broad question, but yeah, if you can comment on that.
No, it's a very good question. I understand the interest over this, but I doubt that I can answer this. First of all, I'm not the person who develops this. It's my colleague who sits right next to me. So I could ask him if I were in the office, but now I'm at home. But the second thing, I think it's going to be published soon, the whole things. They are close to finish the paper. So I guess we all know the answer to this question in the shape of paper, academic paper. It will be a lot of patients, tens of thousands there. So I guess it will be very representative, and we'll probably see some statistically significant effect on the probably reanalysis of the patients or something like this.
Okay, thank you. There's another question about the whether we can say you can send the participants can send a question to the inbox. I think that we'll think about organizing some sort of a forum for further discussions. And I think that if you have some questions regarding today's meeting that you didn't ask but you think about asking them afterwards, you can easily just write an email to the email address that's used to mail the links. So again, we will transfer the question to German and return back with the reply. So that's actually quite an possible way to ask the questions. Okay, so then there's another question in the chat. So the question is, have you observed that relatively simple ranking tools show a much less pronounced improvement in report analysis time than the LLM-based solutions? Yeah, so I think that I think that The question is actually in the time, whether the time gain is really that like high with the LLM based solutions. But I think that maybe the actual time difference is not in the actual ranking itself, but in the fact that the simple methods like that use the scores, etc., require human oversight more than LLMs. Or what do you think about that?
Yeah, it's also ranking. So if you go 1, 2, 3, 4, you're checking the most of them. So if the causal variant is lower in ranks, you will check a bit more variants. So you definitely will spend more time because you will look at more data before coming to the conclusion. How much? I would say evidence model, like this simple model with the scores, it also performs quite well, but not well enough to how to say to provide really fast diagnosis. So we're still like a patient may wait a month until the genetic analyst will have finally time to go through. It's really rare, but it happens. So in this case, even 10% of reduced time would matter a lot because it will accumulate the whole thing.
Maybe I can follow this question up with something from me, because I think that back in the days when there was when there were no LLMs available, still there were companies that actually have suggested some frameworks or like tools that would do genetic analysis and give you essentially the diagnosis, the final causal variant, without even giving you opportunity to look through the candidates. I think maybe some people who are here can recognize the company that I'm talking about, if you know. But still, I believe that there was quite a lot of debate about this back maybe like 6, 7, 8 years ago. Yeah, but with the LLM development, I think the question of sort of trusting the black box interpretation will be probably even more problematic. So now I think that the question that I have is whether you think actually that there will be a time where LLM output in terms of variant interpretation will actually be used without human oversight, pretty much the same way that we in many places. So we see that people don't do Sanger sequencing validation of NGS machines anymore. So could you see something like that happening with variant interpretation, that people will just like think that whatever LLM has found, provided that it actually suffices all of the criteria, that we can not actually look into the raw data itself, but just use the sort of the found variant as the true cause.
It was a very good point. The comparison with Sanger is like straight to the point. It's exactly the comparison which occurs in my brain too. Because previously we were validating all the variants and now we suddenly don't, because it's just pointless to send all of them and spend money on this. And I guess the same will happen with LLMs, but there is a problem with them. If I'm not mistaken, the performance of the models is not linearly dependent on the number of parameters. And I think it can be even asymptotic, the whole performance. So maybe the whole thing will not deliver in the end exactly outperforming human-like performance. Even humans are making mistakes. So the report which a human writes, it's not going directly to the patient or to the doctor. It goes through 2 more stages actually. It goes through like the head of the group and then to the director of the institute, and they at least briefly they read the whole thing. So currently, I'm rather pessimistic about this, and what will happen in 10,000 years, I probably will not see. So in my age, I think maybe it will not be as independent.
Okay, thank you so much. Then maybe if you let me, I will actually, as I announced in the beginning of the meeting, I will actually show a couple more cases which I personally have bumped into while we were doing some of our research, just to add a little bit info to the picture. And yeah, German actually said that his presentation was more about his experience rather than the journal club. And here I will add more of a like journal clubbish part, but still just a little illustration about what the field appears to look like in some sort of systematic search. Let me just show briefly the part of the presentation about that. Just have a short description of what we've actually done. So actually, this is not a published work yet, but I hope that sometime it will be. So there was a colleague of mine who has been doing the review of like nearly 200 papers that have been automatically searched on the web using all sorts of keywords about genetic analysis and generative AI. And there's actually quite a large continuum of papers in the event, at least for the abstracts, and in many cases for the full text, to try to figure out what they're trying to do. And this again is specifically about the generative AI and the large language models for human genetics. And they've categorized these articles into various groups. Not all of the groups are shown here, but this is just a brief illustration of what the groups are. So I think the largest number of papers that we found actually corresponds to the area which we call knowledge navigation and literature review. And this is actually quite an important and quite a common application of large language models. And probably you've heard maybe some time ago there was also a so-called AI call scientist developed by Google, which has also been presented as a companion for researchers to do the literature review. You see quite a lot of application of generative AI to extracting information from literature and actually building knowledge graphs or building some simpler databases of connections between, for example, genes and diseases, genetic variants and diseases. And we see actually that this is nearly a quarter of all the articles that we've reviewed actually fall into that category. So that's a very large field of how people do apply LLMs in human genetics. Then there are other ways how people do that. So there is an area of actually analysis of patient medical histories, 11 papers of that kind. Then there is quite a large amount of papers to do the analysis of medical images. I think some of that has been already mentioned by German, but I will show another example of this later. Then there are tools that are used for variant interpretation, and this is basically what German has shown extensively in his talk. And then there are other areas such as clinical report generation. Again, here a decent number of like 10% of all studies actually talk about this. And then there are other educational purposes, in which there are also actually quite some studies which discuss uses of generative AI in education, in human genetics, and in medical genetics in particular. And again, I'm not trying now to do the extensive review, the extensive literature review here. Just a categorization of what people do apply the LLMs and generative AI tools for in human genetics. And I'll follow with some examples which I just find really nice, and I think they complement what German has already shown. So one example is the tool which is called PhenoGPT, which is actually a tool published back in 2023. So that's already not probably as state of the art as many things in the field are, because actually if you try to search for studies that are using generative AI for genetic analysis, you'll find that the majority of these studies actually are published in the last year or 1.5 years. So this is there is a tremendous growth in the field, obviously because the models themselves have matured and become more sophisticated. But again, this, despite being an older paper, actually I think illustrates quite well what the idea here is. So the idea here is that you have a lot of medical records of the patient, and these medical records, in many cases, they are unstructured. There is a lot of textual information in there. And one of the things that you may want to do is actually what German has alluded to in his talk, which is to actually pinpoint the specific phenotype descriptions in the patient's medical history. And of course, the ideally you should convert these to these human phenotype ontology terms, HPO terms, which are standardized phenotype description in its tree-like structure, which German has mentioned during his talk. And like the PhenoGPT is actually one thing that does exactly that. So it pinpoints the phenotypes from the patient's medical history, and there are actually other tools which do pretty much similar things of detecting standardized phenotypic descriptions in patients' medical history. So that's, I believe, also a good field of application of language models. Again, it's not directly bioinformatics. It's not about genetic variants and variant calling yet, but still it does quite a lot in terms of helping to actually formalize the phenotype description, which is very important when it comes to medical genetic analysis. It's very important to have a good standardized description of the phenotype, because it really matters a lot to have it. Now, besides that, again, this is just one area of which I thought it was rather important to highlight. I've mentioned that there was quite a lot of papers which do all sorts of image analysis, and this is of course not large language models, but pretty much the same architectures and the same type of neural networks are used to solve a variety of tasks. And there are, of course, methods that work with, for example, facial images, also trying to sort of convert it to phenotype descriptions, etc. But one area which we were quite surprised to find when we were doing this literature review was that there's actually quite a lot of studies which are doing a very interesting thing, which is to use the slide images to actually predict, for example, mutations which may be observed in the tumor, which may be driving the tumor. So this is the cancer biology, cancer genetics, oncogenetics part, which is pretty interesting again in terms of pinpointing where to find where to search for the causal mutations. And yeah, this is just one article of that kind. There was actually quite several articles which do pretty much similar things with the slide images, integrating them with the network of information about genes, which is extracted from literature, and combining the two in order to actually give predictions about which genes may be expected to be mutated in a particular sample. So this is also, I think, quite an interesting application, and there are similar applications, for example, to chromosomal abnormality analysis. So there is quite a lot of research there in this multimodal sort of data integration. And also related to images, the thing that I also wanted to show, this actually is related to what German has presented with this Gestalt GAN method, which generates images with the patient images given set of features. And actually, BIF also came across an article recently, which has evaluated the usage of such generated images in educational purposes. Because besides just showing things in for example, in workshops, when you do not want to share the real patient image, but you want to sort of provide the material for the exercise. The same idea can be used to train clinicians, essentially to recognize the syndrome by looking at the patient. And this is a study which evaluated whether these synthetic generated images are actually kind of useful for training the specialists to recognize the syndromes. They've used two syndromes which they were actually training people using their real images and textual descriptions, or textual descriptions and generated images. And they've actually shown that, well, that it varies from syndrome to syndrome, but actually the performance of how people are trained to recognize the syndromes in many cases, generated images perform similarly when compared to cases when people were trained using real images. So this is another sort of interesting use case of how these images can be applied to sort of you generate the images using the trained artificial neural network to actually help natural neural network better recognize the patients, and it actually works, even though the performance actually varies from specialist to specialist. And yeah, this is actually also one thing which I personally was pretty interested to see how these images are applied and whether or not they actually perform similar to real data. This is just an illustration of what people use these kind of methods for in this educational domain. Yeah, that there's a potential to be for these materials to be used in training the medical experts. Yeah, and yeah, this is just again a few examples which we've seen in our this review. Again, I hope that it comes out in a short period of time. And yeah, that's what I wanted to add from my part. And I think that to conclude today's seminar, I would ask my final question to German. The final question will be, so I think that you've already said a few things about that previously, but still I'd like you to maybe elaborate a little bit whether you actually think that there will be a replacement happening, in particular in the clinical genetic analysis and medical genetic analysis. So whether or not there actually will be less human labor involved in the upcoming few years, and if so, by what degrees? Do you expect it to be like really significant in terms of a decrease in the sort of the demand for bioinformaticians, for the people who interpret clinical data, or do you think that it will actually stay pretty much the same? Do you see a change coming in there?
I would say regarding the genetic analyst, there's a huge deficit of these people, and the LLM can kind of improve the situation. But I doubt that someone will be fired because the deficit here now is really big, and yeah, I doubt people will be losing jobs in this specialty because it's very rare and very needed specialty. But bioinformaticians, I'm not so sure. Bioinformaticians are kind of yeah, there are more of us, I guess, and we don't do rocket science really, so maybe some people will be losing jobs, but I doubt at our place. We are there, we are responsible for important parts, and simply firing someone would be difficult because everyone is doing important functions. Someone is responsible for example, long reads, and you ask ChatGPT to write a pipeline for long reads, and it does, but then there are issues, of course, and the people will be coming and asking like why it's working this or that, and there should be human who will be changing this. You cannot always ask ChatGPT to rewrite the pipeline in exactly the way the medical doctors find it useful, find it comfortable for them. So maybe there will be less bioinformaticians need, but I don't see how in our particular situation it can affect anyone. I hope that I think everyone hopes this, that it will just lead to better work, to better research, and not to lead to necessity to fire someone. I think we just can use it as instrument and not replacing someone with this, but vice versa, improve our work quality. So do just things better. But bioinformatics and medical genetics, I'm quite positive. I think it's very high-skilled professions, and they're in high demand. Maybe not all bioinformaticians, again, maybe in some like very industry positions where you just write pipelines. I don't know. Don't take like, I'm not disrespectful toward people who write pipelines. It's very important, I think. But maybe they will be replaced. I don't know. In our institute, I don't see the opportunity to replace many people with LLM.
Okay, thank you so much for your opinion on that. And thanks for everyone who has joined us today to hear this discussion, to hear this the wonderful presentation from German. I hope to see you all next time when we're going to be having a discussion of gene expression data. We'll have actually two expert speakers discussing both bulk RNA-seq, I hope, and hopefully the single-cell RNA-seq. At least that's the plan. We have actually planned to have a second speaker today, besides me as a moderator, but unfortunately he had very important personal circumstances which actually didn't allow him to join us today. But we hope that next time we'll be full squad will be here to have a productive discussion of functional genomics. Thanks to everyone for joining us. See you next time. And if you have any questions which you didn't ask in the chat and you still would like to ask again, feel free to contact us at email.